So our next speaker, who, because of health issues, could not travel in the last few days, is Dr. Ronald Kahn. Dr. Ronald Kahn has had an amazingly prestigious career and is recognized as one of the world's great experts in diabetes, obesity research. He's been has made major contributions in the area of insulin signal transduction. He is the chief academic officer at Joslin Diabetes Center and the Mary K. Ioka Professor of Medicine at Harvard Medical School. He's also served as research director of Joslin for many years and was president of the Joslin as a whole from 2000 to 2007. He's received many awards, such as the Wolf Prize. He will address us now on, we just have to get the title, The Discovery of Insulin, the Nobel Prize, and how it stimulated research for over 100 years, presented by Professor Ronald Kahn. Welcome, Ronald.
Yeah. Okay. Can you hear me all right there? First, let me thank you, John, for inviting me to participate in this marvelous symposium, and also for the great talks by Dr. Norby and Dr. Wright. I want to give a little background even further back than the two first talks, and then try to bring this through the clinical development as well as the research of present day that this has stimulated. So the brief history of diabetes, for those who don't know it, is it's really a very ancient disease. It was first described in Egypt in the Ebers Papyrus in 1550 BC, and was subsequently rediscovered in India, in China, in Greece, and finally in Europe, where Thomas Willis is given credit for being the first person to actually taste the sweet urine of the diabetic patient. But one of the things that's really striking to me in this history is the number of young people that contributed to significant discoveries. For example, in 1869, Paul Langerhans, who at the time was only a 22-year-old German medical student, described the structures within the pancreas that became known as the islets of Langerhans, which contain the insulin-secreting beta cells. In 1889, Minkowski and von Mering recognized that dogs would develop diabetes after removal of the pancreas, and they postulated that the pancreas was a site of production of an anti-diabetic factor. Interestingly, insulin was not the first hormone discovered, although many of us here would like to have that distinction. But in 1902, Bayliss and Starling in England already developed the concept of internal endocrine secretions and discovered the first hormone, which was secretin. And then there were many attempts before Banting, Best, McLeod, and Collop, including important work by George Zoiler in Germany, Ernest Scott, Israel Kleiner in the US, and Nicolae Paulescu in Romania, who were all making really serious and meaningful attempts to isolate the active and anti-diabetic factor in pancreas. As we have already heard from the first two presentations, however, the key breakthrough occurred in Toronto in the summer of 1921. Fred Banting, a 29-year-old orthopedic surgeon having trouble establishing his practice in London, Ontario, had read an article in the Journal of Surgery, Gynecology, and Obstetrics about the relationship of islets of Langerhans to diabetes and to a form of pancreatic lithiasis. And it was this that stimulated him to look for the idea that in dogs, maybe after ligation of the pancreatic duct, you could discover this insulin hormone. And so he returned to the University of Toronto, which was his alma mater, and he convinced the then 45-year-old professor of physiology, J.J.R. McLeod, who we've already heard a lot about, to provide the facilities and several dogs to try some experiments. He also got the help of Charles Best, who at the time was a 22-year-old pre-medical student looking for a summer research experience, to join the laboratory. and the use of the lab and a number of dogs. And he's pictured here in this famous picture of November of 2021, rather, of 1921, with the picture of dog 33, Marjorie, who had been kept alive for 70 days using a crude pancreatic insulin preparation. What's most remarkable about this history, which maybe hasn't been focused on quite as much in the first two talks, is how quickly it moved from this animal experimentation to the patient. The key experiments done in August, September, and October of 1921 were followed really almost amazingly quickly by the first human trial in January 11th, 1922, when Leonard Thompson, a 14-year-old boy who had uncontrolled type 1 diabetes and multiple skin infections, received the first injection of 15 milliliters of crude extract of beef pancreas in the Toronto General Hospital. Unfortunately, there was minimal effect on his blood or urine glucose, and this had no effect on his clinical status. In fact, a sterile abscess resulted. And so enter really, in my opinion, the key person to make this a successful experiment, and that was James Collip, who at the insistence of MacLeod, this trained biochemist, was added to the team to improve the extract. And with this improved extract, Thompson had immediate response with his blood glucose becoming near normal, ketones disappearing, and obvious clinical improvement. So this was the first proof that an extract of pancreas could reverse, at least briefly, the diabetic state. I would comment somewhat ironically that although Thompson went on to live 13 more years taking daily doses of insulin, he died of bacterial pneumonia at the age of 26 because penicillin had not yet been discovered. From the US perspective, this was quickly adopted into therapy. In fact, the first patient to receive insulin in the US was James Haven, who had developed diabetes in 1914 at the age of 14, and who had survived to 1922 through this severe carbohydrate restriction diet called the Allen diet. And he was the first patient in the US to receive insulin. He got it in May of 1922 because his father, who worked for Eastman Kodak, was well connected with a physician who had contacts in Canada. And from a perspective of Boston and the Joslin Diabetes Center, we always were proud that Joslin was also an early adopter. And the first Joslin patient to receive insulin was a nurse named Elizabeth Mudge. Her diabetes had been diagnosed in July of 1917. She also had survived 5 years on the Allen diet. But by August of 1922, she was down to 70 pounds in weight. And when Joslin got the insulin on August of 1922, he was actually apparently so nervous about this that he asked his associate, Howard Root, to give her the first dose, which was quite successful. And after 6 weeks of insulin therapy, she regained much of her lost weight and eventually could walk 4 miles a day and live for 25 more years. Joslin was an early and big adopter. And in fact, this article that appears in the Journal of the American Medical Association, published in June 1923, i.e. before the Nobel Prize, is entitled "The Routine Treatment of Diabetes with Insulin." It may not have been routine for everybody, but for Joslin, he had already treated over 120 patients, even at this time that there was no true commercially available insulin. And we've all heard the next part of this story, the story of the Nobel Prize in October of 1923 to Banting and MacLeod. And just to pick up a little bit on the history by Dr. Norby, that there were some other significant prizes that year as well, including to Millikan for physics and to Yates for literature. What is also very important to keep in mind is how quickly this moved from this experimental discovery to commercialization. By 1923, literally the year of the Nobel Prize, 2 years after the initial experiments, commercial production of beef pork insulin began with Eli Lilly, very soon after with Connaught in Canada, and then soon after that with Nordisk in Scandinavia. By 1936, Hagedorn had begun to make modifications of insulin, adding protamine to prolong its action, protamine zinc insulin, then NPH or neutral protamine Hagedorn insulin. And of course, we know that now we all use recombinant insulin made in bacteria. This was actually first developed in 1979 by Genentech and commercialized by Eli Lilly in 1984. Novo Nordisk made semi-synthetic human insulin to compete. And there have been many now synthetic insulin analogs, insulins designed for specific uses like insulin pumps and closed loop systems. And the discoveries continue. And I'll just add to the kind of recent work that just a few years ago, my laboratory at Emerald Tindus, working in my laboratory, discovered the first insulin-like peptides in viruses. And these actually have unique antagonist or agonist effects, both for insulin and IGF-1. There are clinical trials ongoing of orally active insulin, and their clinical development of glucose-sensitive and other designer insulins. At the basic science side, as was already mentioned by Dr. Norby, insulin was not only self-awarded a Nobel Prize, it was a pace setter of the shape of modern scientific research and many other Nobel Prizes. In 1958, Fred Sanger received the first of his 2 Nobel Prizes for sequencing of proteins, and the first protein to be completely sequenced was insulin. In 1969, Dorothy Hodgkin, an X-ray crystallographer, was awarded a Nobel Prize for unraveling the structure of vitamin B12. But in addition to vitamin B12, with her colleague Guy Dodson, she also solved the first crystal structure of insulin. In 1977, Rosalind Yalow, and had he survived, I'm sure, with Saul Bersin, received a Nobel Prize for the first radioimmunoassay development. And the first radioimmunoassay to measure a hormone was developed for insulin levels in blood. And finally, as also previously mentioned, in 1980, Wally Gilbert, Paul Berg, and again Fred Sanger were awarded a Nobel Prize for recombinant DNA, and the first recombinant therapeutic protein to be given to humans was indeed insulin. So insulin has stimulated and developed over and over again this pathway for great science and important scientific advances. It's important to note that when Banting, Best, McLeod, and Collop developed insulin, or at least first extracted insulin, neither they nor anyone else had any idea of what insulin was at a chemical level. It was simply a biologically active extract. It was not until 1926 that the professor of physiology at Johns Hopkins, John J. Abel, suggested that insulin is likely a peptide, and this was based on his ability to crystallize insulin and the fact that if you treated it with proteases, it was destroyed. Attempts to find insulin receptors also date back to this earlier period, 1949, particularly the work of William Stady at the NIH, who tried to do binding studies with insulin unlabeled and then labeled. These were very controversial. In fact, Bersin and Yalow felt that they were all artifactual, and so these fell into disfavor. But this idea that insulin must act at the membrane of the cell was further stimulated by the work of Rachmael Levine and his colleague Goldstein, who demonstrated that insulin stimulated glucose uptake in the dog. And so this really focused on the membrane as the site of insulin's first action. And it was in 1971 that Pierre Fauché, working in Jesse Roth's lab, and Pedro Quattrocasas in another lab at the NIH, were the first two to identify insulin receptors by specific binding to liver plasma membranes. And here you see this kind of classical experiment showing on the left insulin's different categories of insulin's ability to compete for binding to this liver insulin receptor. And on the right, the biological activity of these insulins, which completely parallels their binding affinity. Very quickly, we recognize that insulin receptors are conserved across species, all the way from mammals all the way down to C. elegans, that there is insulin-like growth factor 1, IGF-1, which binds to its own closely related receptors, but these two receptors can bind to each other's receptors, creating some spillover in their signaling and physiologic activities. That insulin receptors are present on all cells of the body, including tissues not previously considered insulin sensitive, like lymphocytes and brain. And these two concepts are becoming increasingly important with the link of insulin resistance to Alzheimer's disease. And finally, and most importantly, that insulin receptors can be regulated or downregulated in diseases and contribute to insulin resistance. This actually provided the next step in understanding insulin action, which was using antibodies from a patient with insulin resistance. Masato Kasuga, Anders Carlson, working in my laboratory, discovered the fact that the way insulin signals is through the fact that the receptor itself is a tyrosine kinase. And here you see from some 40 years ago, this experiment in which we took cells in culture, labeled them with radioactive P32, immunoprecipitated the insulin receptor, either in the absence or presence of insulin stimulation, and saw autophosphorylation of the insulin receptor's beta subunit. The full structure and sequence of the insulin receptor is now known, and you see that on the left of this slide, as well as a three-dimensional reconstruction of the extracellular domain on the right. And this, a beautiful model of the insulin receptor binding the insulin molecule shown on the receptor in that red and blue structure, activates the kinase and creates a signaling cascade responsible for all insulin action. The first step in this action, both for the insulin receptor and the closely related IGF-1 receptor, is to phosphorylate some intracellular substrate proteins called IRS proteins, shown here as IRS 1, 2, 3, and 4. These then lead to a cascade of phosphorylation reactions through PI3 kinase and through AKT that ultimately stimulate glucose uptake into the cell, lipid synthesis, glycogen synthesis, inhibit gluconeogenesis, stimulate protein synthesis, and acting through a separate series of pathways, stimulate growth actions like growth and differentiation. So insulin action at the cellular level is extremely complex and has multiple steps, but we really understand now many of these steps that are critical for muscle, liver, and fat actions of insulin. The other thing that's really become well recognized is that for type 2 diabetes, which was actually alluded to in those after dinner comments, I guess, by Banting, associated with obesity, the type 2 diabetes is really not due to insulin deficiency, but rather primarily driven by insulin resistance. And insulin resistance is at the core of many metabolic diseases that are associated with the metabolic syndrome, including dyslipidemias, central obesity, hypertension, and accelerated atherosclerosis. And we also know that insulin resistance is a major component of non-alcoholic fatty liver disease called NAFLD. It's a essential component of reproductive dysfunction in women related to polycystic ovarian disease. It occurs, when it occurs, it increases risk of a number of cancers. It causes accelerated Alzheimer's disease and ultimately impairs longevity. So the real challenge for the current time is not insulin deficiency, but insulin resistance instead. And this can have many drivers from genes and the environment and epigenetics and the microbiome. One of the important questions of our laboratory now and at this time is, what are the real drivers of this insulin resistance? And are some of them intrinsic to the cell? Do patients with type 2 diabetes and metabolic syndrome have genetic or epigenetic factors that already drive this insulin resistance? And I won't have time to go through a lot of the current work, except to say that it is possible to make iPS cells. They can be reprogrammed from muscle or skin biopsies or blood cells using viruses like sendivirus, and can be differentiated into all the kinds of cells of interest for the study of diabetes, like muscle, liver, fat, beta cells, brain cells, etc. And using these cells and patients with different levels of insulin resistance, either severe due to an insulin receptor mutation, or type 2 diabetes, or even insulin resistance without diabetes, we can take these iPS cells and make iPS-derived muscle cells, myotubes, or liver cells, hepatocytes, and we can study each of these for insulin signaling, glucose uptake metabolism, protein expression, and mRNA expression, and signaling through phosphoproteomics. And remember that picture of the insulin signaling cascade? Well, what we find is that when you take cells from patients with type 2 diabetes and you look at this insulin signaling cascade, many of these sites of insulin's metabolic actions, the stimulation of AKT and enzymes like glycogen synthase kinase, transcription factors like FOXO, and glucose uptake. If you look at the cells from the type 2 diabetic compared to the control, although they stimulate, for every one of these effects, they have reduced stimulation compared to the control. They have insulin resistance in this tissue culture dish model. And we can actually try to get our hands on what are the molecular drivers of this insulin resistance, not only using phosphoproteomics, but using proteomics and gene expression. And when we do that, we find that there are many, many alterations. This is what's called a heat map, where blue means a low level of activity and red means a high level activity. You can see at the top the insulin regulated phosphoproteomics, but in the middle, you see type 2 diabetes changes, where in this case, there are sites that are more phosphorylated in type 2 diabetes than in the controls, and another whole group of sites where they're downregulated in their phosphorylation compared to their controls. And using this, we can begin to look back at this complex insulin signaling map with the receptor and the substrates and all the downstream intracellular effects, and we can find these common sites of alterations, phosphorylations of IRS1 or AKT or FOXO proteins, or proteins involved in RAC and ROS signaling, or even proteins involved in the nucleus for gene expression, DNA, and mRNA splicing and processing. And so this provides the new roadmap where we can begin to think about how can we use this information to develop new screens, such as drug screens and CRISPR screens, to reverse the insulin resistance of type 2 diabetes, something that isn't itself treated with insulin except by increasing the dose. So in closing, I would say the discovery of insulin is not only about the past, it's looking to the future. Since the discovery of insulin, there have been tremendous progress in making better insulins and more sophisticated and physiologic delivery systems. These include glucose-sensitive insulins and viral insulin-like molecules with unique properties. Over and over again, studies of insulin and diabetes have provided models for studies and advancement in many other fields, as was shown in this talk and in the previous talk as well. And finally, insulin resistance, which is the primary driver of type 2 diabetes and metabolic syndrome, remains the challenge of this century. New tools like iPS cell-derived models of muscle and liver from these patients mirror the defects of insulin resistance in vitro, and this provide a key tool for the future advancement and development of new therapeutics. So thanks for your attention and allowing me to participate in this symposium.
Thank you, Dr. Kahn, for this impressive and elegant talk and showed the tremendous progress made over the last centuries.